And why would we want to talk about methods? Well, so far we've not really talked in detail about particular studies. We've not talked about particular sets of experiments. We've sort of talked about things broadly, right, Colin? We've not really said, like, here's a specific study where, I don't know, they poked this animal 12 times and then it jumped once, and so there we know something, right? So there hasn't really been a need for you to understand methodological approaches at the moment. But as we go into the rest of the semester and deeper into the book, you will have studies that are sort of advanced designs, right? I'm not expecting you to know all of this, right, Eric? So you don't need to like walk out of this class today and be ready to design any sort of behavioral neuroscience experiment ever. But just so you have an understanding of what are some of the limitations, what are some of the answers that we can get from these particular methods, I think that's going to be helpful. Right. So we're going to talk about something called ablation. How many of you remember Pierre Florence? He's your hero, right? Yeah, right? I mean, you've got like a Pierre Florence tattoo, I can tell. Of course. On my shoulder. Yeah, on your shoulder, right? Yeah, yeah. That's a great place to have one. So we're going to talk about ablation. Remember, he had that idea like, hey, if I like scoop parts out of a brain and then you can't walk, well, that must be the part of your brain that makes you walk. Right, Jason? So that makes a lot of sense. That's sort of crude and not probably the most detailed approach. We're not going to learn a whole lot from that, right? But we've got much more precise techniques that are available now, right? And so when we're going to do ablation, that's going to work a lot better for us. We're going to talk about recording and stimulating neural activity. I know we were sort of snowed out the day that I was going to tell you all about Hodgkin and Huxley, right? But I'm sure you listened to that lecture and it was brilliant, right, Eric? But Hodgkin and Huxley are really important. That's Alan Hodgkin and Andrew Huxley. So for those of you that are Brave New World fans, that's his half-brother, right? So there you go. There's some Huxleyness for you. So we'll talk about recording and stimulating neural activity, what we can learn from that. We'll talk about some neurochemical methods, and then briefly we'll talk about genetic methods. Somebody's going to have to remind me about that because I don't have a slide for it. But I'm going to tell you all kinds of cool stuff about something called optogenetics. It's going to be a blast. All right. Experimental ablation, this is producing a lesion in your brain. All right. Not something you want to do on a normal basis. Okay. You don't want to go like, boy, guys, let's go out and get us a brain lesion today. Regan, that's not a great thing to do on your Saturday night. There are much better things you could do, like get a Pierre Florence tattoo, right? It's a much better use of your time and money. There are a couple ways that we can go about doing this. One of these is using radio frequencies. So there you go, right? So just like crank up your radio next time you want a brain lesion. It doesn't exactly work that way, but you can use radio frequency. What's more common is using what we call excitotoxic lesions, right? How many of you remember that discussion about glutamate? Okay, we talked about glutamate being that excitatory neurotransmitter. Later, when we talk about seizure disorders, we'll talk a bit more about glutamate. We'll talk about how that could lead to cell death in seizure disorders, right? If those cells become overexcited.
Would MSG increase your glutamate?
Yeah, that's actually a great question. You know, a lot of folks get really worked up about MSG, right? Like, oh no, don't give me MSG. It's probably a good idea to avoid it, more for the S part, which is the sodium, right? Because you don't want to jack up your sodium levels too much. But glutamate, I don't know, you'd have to really, I think, eat a lot of food with him. You'd have to, like, before it's going to cause you any sort of neuronal problems. Well, that's a possibility. I mean, if you're constantly eating food with MSG, I don't know. I think you're going to die of a heart attack first. That's just my sort of feeling on that. I'd be more concerned about the cardiovascular aspects. But that's a good question, Colin, actually. So, you guys familiar with MSG? It's monosodium glutamate. So food additive in a lot of foods. But yeah, the sodium's the more frightening part on that. Because you also have to worry about like how many receptors do you have, right? So you're not necessarily increasing receptor numbers either, right? Just because you're increasing sort of systemic glutamate, not all of that's even going to make it to your brain, right? We have to think about things getting across the blood-brain barrier, being broken down in your stomach. So I don't think dietary MSG is going to really run you into any sort of neurological problems. So there you go. I don't know. Is that a debate you were having with someone? I mean, it's supposed to be.
Not really.
Not really. Okay.
But that's a good answer.
Yeah, well, okay. Well, there you go. Now you can have that debate,
Yeah.
right? You'll know which way to come down on. So quite often, what they'll use is a glutamate agonist. Okay, so if you use a glutamate agonist, remember we had that whole agonist-antagonist discussion, right? But if you use a glutamate agonist to increase glutamate activity in your brain in a very localized region, right, that can kill a few cells in a particular brain nucleus, in a particular circuit. For how many of you are psych students? How many of you took that experimental design class? Most thrilling, wasn't it? I didn't teach it, so it probably was actually pretty good. I taught it once, Eric, and they said, I don't think we need you to do that again. I was like, I didn't think I needed to do it the first time, but you made me. So there you go, that's what you get. Right, Jason? It's like when people listen to me and they walk into the wrong class. So, you have to have a control group. We're always going to have to do a sham lesion in these studies, right? How many of you have ever rammed a metal rod in your brain? Yeah, nobody, right? Because it's not a bright idea. So Jasmine, anytime we make one of these lesions it's going to be a non-human animal, right? So it's going to be monkeys, rats, mice, something like that. We are going to be injecting something into the brain. So we're going to have to put a syringe in there, right? And that syringe is going to take up some space, and that's going to cause some damage going in and coming back out, right? And so we don't want the results of our studies to be just because we jabbed the brain with a metal stick. So we have to do a sham lesion as well, too. Usually we'll just squirt in some saline, which is really just salt water for any of you who, I don't know, anybody drinks saline on a regular basis? Nobody does that. How many of you put saline in your eyes or up your nose? There we go, right? I know some people do. How many of you use like contact solution or eye drops? Yeah, it's really just saline, right? So there you go. So we're going to use saline. It's perfectly safe to stick in your brain. Again, I wouldn't recommend it, Ray, because that metal rod can cause some damage. Don't worry too much about this slide. That's just showing you that there are some lesions. You can see right there next to that arrow, sort of a white spot. That's where the cells have been killed. Later, they're going to be processed with something called a Nissl stain. It's going to turn the cell bodies kind of purple, so you can see them in a microscope. And you can see there's clearly, right in that area, there's sort of a white spot where there should be these nice little purple dots, but they seem to be absent because someone did a nice excitotoxic lesion. How many of you have ever tried to hit something in the brain? Okay, that's what I thought, right? But Abby, what position do you play on the softball team?
Center on the field.
Okay. What's really great is when people stand still so you can throw the ball to them, right? Okay. So they're going to be anchored somewhere and they're not going to be moving, right? So if you're trying to throw into second or third base, for example, that base doesn't move, right? It's anchored where it is. It's probably like a peg, right, in the hole, and that's how they hold things. Same thing if you're a pitcher, right? They don't move the bat. The batter's box isn't constantly moving, right? That would be really difficult for the pitcher and the batter, I would imagine, right? And you're trying to constantly move around. It'd be kind of like that if you were doing brain surgery or trying to do an excitotoxic lesion. If you didn't fix the head somewhere, right? If you didn't hold the head still, can you imagine like trying to, you got a syringe in one hand and somebody's head in the other, and you're just trying to, doesn't seem like that's really a great idea, right? So we do this thing called stereotaxic surgery. We use a stereotaxic device. It gives us this nice sort of 3D coordinate system so we can locate parts of your brain, right? That's how we know where things are. We're going to use this device. It's going to hold, if it's an animal study, it's going to hold the animal's head still. Not too big of a deal. We have these things called Stereotaxic Atlas. This is just a map that someone's created of that particular species brain. So you can get a rat brain atlas. It's really a thrilling read. So you just, you just flip through these pictures of slices of a rat brain, and then there are all these arrows that tell you what the brain regions are called. And if you want to hit one of those, you just find the coordinates, 3 sets, you know, X, Y, and Z, and you drop your electrode or your syringe right there, and you're in it. I mean, give or take rat brain variation. This is a picture from a stereotaxic atlas, just to give you an idea. It's marked off in grids. Let's say you want to hit a particular area. This is called the fornix. Doesn't really matter what it's called, but you can find that location. There's kind of your X and Y if you want to know your sort of Z-plane. It'll actually have a number printed down here at the bottom corner. The device that's going to hold the animal still is called a stereotaxic apparatus. Again, this will hold the head still. It'll hold the electrode or that syringe that you're putting in still, so that you're not kind of, you know, trying to hit this moving target. Okay? It's not dodgeball. Right? Desmond, we're going to hold everything still and in reference. Here's what this looks like. So this is a drawing. This is a rat. For those of you that don't know what a cartoon rat looks like, that's it. These are knobs that you can use to adjust the position of this electrode. Not while it's in the brain, that's a bad idea. You want to pull it back up and out and then move it around and then drop it back down. Have you ever tried to push something through your skull, Emily? Great. Not. Yeah, that's probably going to be a waste of time. So what we have to do is we actually have to make a hole right in the skull and then put the electrode in. So just heads up there. Here's one if you're doing it with a human. That looks, I don't know, uncomfortable to say the least, right? I don't know if anybody wants to sign up for wearing one of those around. But it's still important. It's kind of the same idea. You want to find how best to get into whatever brain region you need to target. Maybe you need to remove a tumor. In humans, that's typically what you're going to be doing. All right. Has anybody ever held like an actual brain? I know, Eric, you said you did a sheep brain dissection. You guys have done the same. They're fairly firm, right, by the time you get your hands on them. And the reason they're fairly firm is that they've been processed, right? Are processed with something that's like a formaldehyde. Okay. So it probably smelled wonderful, didn't it? No, it smelled awful. It was in some liquid, probably. It was like some liquid around it, right? Okay. It's quite similar to embalming fluid, but this is going to go like really jolly, really fast. I don't know if any of you have ever been to a funeral. I know, right? Louise, I've got to, right? It's kind of like embalming fluid, right? So they kind of preserve the person for a little bit so you can get a peek at them. And then you do the same thing with brains. Otherwise, brains are really squishy. Has anybody ever seen a brain like a fresh one that's not been processed? It's just kind of like squishy, right? So you just kind of just squishes out. So you want to make sure it's firm so you can cut it. So we're going to put it through this process called fixative. We're also going to cut this on a thing called a microtome. This is sort of what a microtome looks like here. It's kind of like a deli slicer, right? Everybody loves deli meat. And nobody's going to get deli meat again, right? Because you're going to think about like slicing a brain on the deli slicer back there at Kroger. Nobody buys deli meat like freshly chopped. Eric did.
Oh, I don't care.
Yeah, trust me, it's not the same. They're not going to let you slice brains back there. I've asked. No, I'm just kidding. It won't make a thin enough cut, so they're kind of useless. When we cut this, we're going to cut something around the like 50 to 100 microns or micrometers, right? So we're talking about something like super thin, okay? Even if you get thin sliced deli meat, it's thicker than that, just as a heads up. This guy's called a cryostat. Some of you may have seen these if you've ever visited kind of labs or anything. It's for cutting tissue. Kind of keeps it cold, so it's frozen, right? So you can cut these sections frozen. That's okay sometimes. Sometimes you don't want it to be frozen. You have to use a different approach. Hey, who loves the color violet? Nobody. That's really sad. I thought there would be some people that really kind of get into that. If you just take a brain and you slice it, you're not going to be able to see anything, right? You're not going to be able to see any of the cells. You're not going to be able to see any of the axons or dendrites. You're not going to be able to see anything at all. It's all going to be kind of the same color, a little different, a little whitish in some places, a little more grayish in others, and that's sort of it. Because we really want to see cell boundaries and maybe even count cells, see if we actually killed some cells, all kinds of things, we'll use this variety of stains. This is one called Creswell Violet. This is a pretty simple, straightforward, sort of non-specific stain that will just stain a bunch of cell bodies. How many of you have heard of gray matter and white matter? Just sort of talked about that, so just tossing that back out there. The white matter is going to be the axons. So if you look here, these are all big fiber bundles, right? So they're big axon tracts. The gray matter is going to be cell bodies, right? And so if you look at these areas that are purple, they're really dense in cell bodies, right? It's not too big of a deal. We have to use different microscopes. How many of you have ever used a microscope? Like sophomore biology in high school, right? They're like, hey, look at these awesome cheek cells that you have. Anybody do that one? Okay, I thought so. That's still like a thing, right? So that's sort of just like what we call a light microscope. It's sort of low level, because things in your brain cells are super small, right? We're not just looking at like a whole cell. We want to look at the parts inside of the cell. It gets a little tricky because how many of you understand wavelengths of light? They kind of go like this, right? There's like a wavelength, whatever. That's okay. So if you think about how many of you can see people? Yeah, people are bigger than wavelengths of light, Elizabeth, just to give you a heads up, right? Wavelengths of light are really small. And so if you are larger than a wavelength of light, I can see you with light. So that works. The problem is when you get smaller than the wavelength of light, and you get smaller than the space here between sort of the peaks, right? Then it becomes impossible, like physically, right, for me to be able, because you're our physicist in this class, it becomes impossible for me to see you because you're falling in between those peaks, right? So in that case, when we're thinking about things like the synapses that we were talking about sort of last week, think about those synaptic vesicles, all of those super small things, we can't see those really with a light microscope, so we have to go with something else. What's smaller than light? Hey, how many of you know about electrons? Yeah, those things are super small, right? And so we can shoot a beam of electrons at brain tissue, and based on the way it reflects and whatever, we get a nice pretty image of the way that your brain, sort of the subcellular parts. So that's pretty cool, right? So that's exciting. The problem with that is nothing's going to be in color because it's smaller than a wavelength of light. You know, wavelength of light is kind of what gives you color. So we're not going to see anything in color. It's all going to be kind of like grayscale. It's just going to be density, right? If it's got a higher contrast or a lower contrast. Not a big deal there. We also have a thing called confocal laser scanning microscopy. That sounds super awesome. I know everybody got really excited about the lasers, right? But they're all kind of in a box and you don't really get to shoot people with them, so it's not as fun as you think. Although you can get some 3D images from this, right? So you sort of like scan the tissue at different depths with laser, and there's like a spinning disc, and anyway, it's not that important, but it'll give you some cool images. This is an electron micrograph. Okay, so that's like super cool. This coming down is a terminal. Remember that thing that kind of was shaped like this? Okay, you can kind of see that, follow that sort of membrane around. These guys that are round and sort of have like stacks in them, those are mitochondria. How many of you are familiar with mitochondria? I mean, those are things, you've got a few of them. So they're there. These smaller circles, try to draw one. Those guys, Those are the synaptic vesicles. These two arrows, trust me, there's nothing in your brain that looks like that. Those are added after the fact, just to add it in anybody get confused. Those are pointing out synaptic clefts. So those two little fuzzy areas that kind of look like caterpillars, those are all of the proteins and the density there at the synapse, right? So that's when the two cells are communicating with each other. This circle here is an air bubble. I don't know why they selected an image with an air bubble on it. That's just, it's bad. Should have photoshopped that out. Would have made me feel better. This is from a scanning electron microscope. This is actually a cell body, and you can see all of the dendrites and connections and so forth. Not too big of a deal there. What's really a fun thing to do in the brain is figure out which two brain regions are connected, right? Because then you can think about what information is being passed from one place to another. Unfortunately, we don't have like a map, right? I mean, nobody uses maps anymore. Everybody uses GPS, right? Jasmine, do you know how to read a map? No, of course not, right? I mean, Rand McNally is totally out of business. No one even, Eric and I were like the only two people who know Rand McNally. I don't know, I used to own a map. So, we have to sort of create maps in the brain because we don't know which two brain regions are connected. We have stuff called neuroanatomical tracers. Really, that's scientific grade food coloring. Sort of what it amounts to. You can put the molecules in one place, you can wait a few days and they'll show up somewhere else, and then you can say, well, those two areas must be connected. Who remembers that axonal transport business that we talked about? Right, where we were taking molecules from either the cell bodies or from the terminals and moving them in one direction or the other. Okay, so we'll take advantage of that for this sort of approach. Some can move forward and some can move in reverse. So there's anterograde, there's also retrograde, which is not on this slide. Don't worry about this, these are just images. Oh, this is kind of interesting. This was, no, that wasn't with herpes. I always like to ask who loves herpes, but nobody ever raises their hand. Most people make that face, Travis. I don't think so. What's awesome, well, not much, but what's awesome about herpes is, I know, right, you're like ready for this. Hang in there. It's a virus, so just to let you know. And the cool thing about a virus is it can get in your cells, and then it can make your cells like do things that they don't normally do, right? Like copy the virus DNA, then they'll explode, and then all that virus DNA goes all over your body, and it does the same thing again, right? And that's kind of how a virus works. Now, if you can cut off the part of the virus that's going to actually cause you disease or illness, Eric, you can use that part that gets in the cell. That part's really awesome, and it's really great for like studying the brain, because that virus can travel through brain cells, and we can track its progress over time and see which brain regions are connected. So that's actually pretty cool. One of the other viruses that's often used is rabies. Anybody a bigger fan of rabies than they are of herpes, right, Chloe? I mean, if you got to pick one, who's going to be a fan, right? I don't know. It's like nobody really wants to. Nobody wants to make that choice, right? It's like, which one are you going to take, herpes or rabies? I don't know, right? I mean, so I think about this sometimes, like what if you're ever in this sort of situation? And it can happen to you, Jason, trust me. And somebody says, I'm going to give you one of these two viruses, which one are you going to take? It's always important to ask which version of herpes. Because there's herpes simplex and complex, right? Get it? Trick question. The one that gives you like cold sores, much better than rabies. Just as a heads up there. Rabies will kill you. You guys know that? Anybody vaccinated? Anybody live with someone who's vaccinated? Anybody have a dog? It's got a rabies vaccine, Emily? Get that taken care of.
My parents deal with it.
Okay, your parents, that's fine. You're okay, you're in the clear. Don't worry about that, that's just a picture. So there you go. This is what happens when you make an injection, you wait, and you see where else you find that particular tracer. a week or so later and you go, whoa, those two brain regions are connected. That's awesome. All right. All of those methods, Matt, required something, mostly a hole in the animal's head, right? And unfortunately, there aren't a lot of people signing up for that. So if we want to know what's going on inside of like a human's brain, like what's going on there, we have to think about ways to do that without cutting open your head. Okay? And I know it really takes the fun out of it, but whatever, right, Eloise? So, how many of you have heard of computerized tomography, CT or CAT scan? Right? There's MRI and fMRI. Okay. We also have PET scans and diffusion tensor imaging, or DTI. This is kind of a weird way that we can figure out where water molecules are oriented so that we can track axons in the brain, which is kind of fancy. So there you go. This is the Stargate portal that we're going to... Anybody see that movie? Yeah, I should share this. My brother, who's like 11 years younger than I am, scared to death of the movie Stargate when he was a little... I know, right? So of course I watched it all the time. Being 10 years older, that's just what I was required to do. I don't know why he was afraid of that. It's not really a frightening movie, is it? I mean James Spader is always creepy, but... there you go. So if we want to stick somebody in a CT scanner, this is actually using a lot of X-rays, so you sort of like a 3D X-ray is really what it amounts to, right? So we're going to go through step by step, piece by piece through the brain and create sort of a number of X-rays that will stack together and make a 3D image. It's pretty good for a lot of things. You know, we can find tumors, if you've got a brain hemorrhage, something like that. Doesn't really give you any sort of activity idea, right? It's just simply, here's a static image of your brain at this moment. There you go, this is sort of a top-down view. We can see in this, let me find my mouse, we can see here eyeballs, these two circles. See them sort of again right here. Here are the ventricles. Remember we talked about ventricles a week or so ago. You can sort of see the lateral ventricles as we're stepping through this person's brain. This is an MRI. MRIs are similar in the information they can give you. So this is obviously sort of a sagittal view of this particular individual. And you can see different parts of their brain, different brain structures, and different steps moving through. fMRIs are cool. Those are functional. They can actually measure brain activity in particular regions. They sort of work off what we call difference images, right? So your brain's always doing something, okay, even if you don't think that it is. So it's important to compare, like when you're doing something like reading to when you're just looking at pictures, because you're, you know, some of the same brain circuits and brain regions are involved in both activities because they're both visual activities, right? So we have to subtract activity from, brain activity from one task from another. This is that diffusion tensor imaging. This has actually been colored to show you sort of where these axons go. Again, this is related to like water molecule orientation. All right, don't worry about this. It's all just recovering everything, or covering everything so that you can have an idea about what's going on. Now, Alyssa, that was just giving us information about sort of structure, right? But we want to know what activity is actually going on. Remember that awesome thing that we talked about for quite some time called the action potential? Okay, so we want to think about that electrical activity in the brain and how we can measure that. We do this with something called microelectrodes. These are very small electrodes. They're sort of little pieces of metal that'll be covered with an insulator except at the tip. We kind of let the tip open so we can measure that electrical activity from individual cells. We have what we call single unit recording. We're going to record from a single neuron. Now obviously we're talking about millivolts, right? Because we were saying like negative 70 millivolts to like positive 30 or 40 millivolts, right? That's the range. So we're going to have to amplify this, right? How many of you know the sort of voltage coming out of the sockets in the wall?
want to see it?
Yeah, like 110, 120, something like that, right? It varies just a little, but that's the range we're talking about. So that's volts with a big V, not with a little m in front of it, right? So we're talking about a millivolt is like a lot smaller than that, right? So for those of you non-decimal system fans, just know that it's a lot less than a volt. And so we have to amplify that so that we can get that up to the point where we can measure it and record that information. You can also record from multiple neurons at one time, right? Sometimes they'll do this with like EEG devices. I don't know if anybody's had an EEG before. Sometimes they give this to you if you've had a head injury, or if you have some other sort of neurological issue, they'll give you an EEG. That's kind of nice because, again, they don't have to cut a hole in your head. And they can just slap these electrodes on the surface, and there you go. There's also this awesome thing called magnetoencephalography. It's a way that you can use the magnetic fields generated by your brain. And don't like try to stick pieces of metal to the side of your head. It's not going to happen. It's not that powerful. But anytime you have electrical activity, you can generate a weak magnetic field as well. And we can measure that with these giant magnets. It's kind of exciting. So this is a rat. Sometimes you want to record from these brain regions for an extended period of time, right? You don't necessarily want to just like stick an electrode in for the day and like pull it back out. So if you can create some sort of semi-permanent apparatus where you can plug in and plug out multiple times over the course of several weeks or months, that's a pretty advantageous technique. They use dental cement or dental plastic, right? It's the sort of, I don't know, pinkish substance they use to like make dentures, right? Sort of blend in with the inside of your mouth. All right, hey, check that out. That's an EEG. We'll talk a lot about EEGs when we get down to the sleep chapter, because EEG frequency is going to really indicate your stage of sleep. Okay, that's going to be important. You're going to get these sort of outputs. Now, again, we're taking thousands of action potentials from multiple neurons and adding and subtracting all those together to kind of make this sort of more complex waveform. We should think about metabolic activity. We can actually measure, and again, we have to sort of get in your brain. We can measure real-time neurotransmitter release in your brain. That's a complicated and delicate process. And so a lot of folks will use techniques where they'll measure sort of activity after the fact, right? So how many of you, this is a good one for you, Eloise. I'm assuming you're familiar with box scores, right? So like in the newspaper after like a baseball or softball game, they'll give you a box score.
Okay.
You're not familiar with box scores?
Not in the newspaper.
Not in the newspaper. You don't read the newspaper? Have you ever heard of one? So it's like sheets of paper, and they print words on them, and you can like, I know they don't make too many of them anymore, right? And you can actually like look at these and learn things. They're kind of fascinating, right, Abby? It's really fascinating. So they have box. It's kind of a game summary, right? Like in this little capsule, and it will tell you the activity of each particular player, like how many times they were at bat, how many hits, if they were walked, or multi-base hits, things like that, right? You can sort of look at that game summary, and you can get some idea about the actual activity during the game, right? If you guys are not like stick and ball fans, what's another sport going on right now is basketball. You can look at game summaries, right, Eric, and you can say, wow, that guy played for, I don't know, 32 minutes, he had 27 points, 3 fouls. You know, they can give you those line items, and you can kind of assume, you know, something about how much activity was expended during the game without actually having to watch the game, right? So kind of a quick and easy way to figure out what was going on and get caught up. We do the same thing in the brain. We can't always just watch each particular neuron or each synapse, right? But we can measure activity after the fact, and we can look at some of those byproducts. And that way we can think about what the metabolic activity was, right? Because we do know that your brain cells are going to increase their metabolic activity if they're increasing neural activity, right? The more action potentials that cell is firing, then the more active it has to be metabolically, right? You're going to be using more ATP, you're going to be making more of those byproducts, and we can measure that. And these are some of the methods we can do this. One of these is 2-deoxyglucose. This is a radioactive label. We can track in your brain, right? So we will give you this injection. Then we'll image your brain while you're doing something, and we can see which parts of your brain are lighting up with the radioactive glucose, right? Because if your brain's using glucose in that particular region, then it must be really active. If it's really active metabolically, then we know there's a lot of neural activity going on. So who loves radioactive sugar? Right? You never thought that was going to be like the topic of the class, right? All right. We already talked about fMRI a little bit. A lot of this is related to blood metabolism changes, not too big of a deal. Don't worry about the FOS protein, but it's the same idea. PET scans, MRI. Oh, sometimes we don't just want to see what you can make your brain do, but we want to make it do things, right? And so we're actually going to inject some electrical current. We can do this. We can inject an electrical current in one region. Now remember, we're using this electrochemical signal, right? So we can inject an electrical current, and that'll cause some downstream effects. The other thing we can do is we can actually insert those excitatory amino acids, for example. We talked about that with the GABA agonist, or the glutamate agonist, sorry. And you can stimulate using chemicals as well. And there you go. There's just what we call a cannula. It's really just a tube that goes into your brain, and you can squirt stuff in it. Cannula, I know, Jasmine, cannula is a fancy word, isn't it? It's a tube. You squirt stuff through it. Not a big deal. Who loves magnets? Anybody collect magnets? Anybody have a refrigerator? That's what I thought. You guys just live out of coolers? I don't know. I looked at buying one of those Yeti coolers once, and I thought I can just buy a refrigerator instead. Because I saw the price tag on one. It's like, forget that. Unless I steal it, I'm not owning one. I'll just go to Lowe's, and you can even buy that one with the picture. I love the one that takes the, I want one, but my wife tells me no. It takes a picture of what's on the inside of my refrigerator, so I don't even have to open the door. Now that's energy efficient, Matt, because I don't, every time you open the door, you know, that's wasteful, right? So if you're like opening the door, like, do I have cheese? You know, if you shut the door and it takes a picture, then you know if you have cheese. It's better for the planet. I don't know why nobody else thinks that. That's brilliant. I think it's...
Isn't it using energy to take a picture?
It's a give and take. There's no difference. It's just illusion, man.
Just if it breaks, it'll cost you a lot more to get it fixed.
Yeah.
But I can probably watch Netflix while I'm deciding what I want to eat. That seems valuable to me.
Would you know when you like make stuff if you're out of it?
Yeah, you know, most people would.
I'm just kidding.
I don't know. I just, this is why my wife doesn't let me go shopping. I just buy things like off the shelf. Like, oh, that looks good, and I don't even know what we have, because I don't. But I'm the one who does most of the cooking too, so I should know, I just don't pay attention. It's not on my list.
Live your dream.
Huh?
Live your dream.
Yeah. Just go buy whatever. Wait until you guys make as much money as I do, you can buy whatever you want at Kroger. That's it.
Gucci Kroger or?
Yeah, I actually lived in that direction, that's why I shot there. Although the one here on 5th Avenue is like stepping it up, right? I mean, they just had that big remodel. So.
It's still not that nice.
It's still not that nice? I don't know. I've not been in there. I go in if I have to, because like if I'm here and I'm like, oh, I need something as an emergency for work, but otherwise, yeah, I go down on 60 in Barbersville.
Okay, build your own six-pack now. That's a step up for us.
Really?
Yeah.
Huh. Something to look forward to. Until they start letting me drink while I shop, I don't know if that's gonna.
There is a Kroger in Ohio that's like that. They have a bar, and like a bar that you can go to and sit at, and they give samples of beer and wine.
I'm just wanting to drive to Ohio.
Jungle gyms.
Huh?
Jungle gyms.
Yeah,
Jungle
the-
gyms?
Yeah.
Yeah, but that's that's a whole different
Oh,
world.
yeah.
That's a whole different world, jungle gyms. All right, so magnets, you can turn your brain off with magnets. I wouldn't, try it on your own, right? But they can use then believe it or not, this is a magnet. This is not a set of binoculars the guy has on the back of his head. These like 2 eye holes back there, I don't know. So you can use this to turn on and off your brain, or particular parts of your brain. They use it a lot for studies. Oh, hey, remember that time I told you I was going to have to add a slide? Apparently I did. Here it is. It's called optogenetics. Guys, this is actually super cool. Who loves rabies? Okay, nobody. I was just going to see if I tricked anybody into getting excited about that. So this is going to use the same sort of idea of viruses, Allie, to get inside your cell and to trick your cell into creating ion channels using different DNA than your DNA. And what's cool about these ion channels is they're actually sensitive to light, right? So most ion channels that we've talked about so far open and close because of neurotransmitters or because of some other chemical, right? These open and close because you shine light on them. Now, how many of you have ever tried to shine light on your brain? Doesn't work very well, right, Travis? Because you got like hair and scalp and skull and other things. But if you're a rat, again, we can put a hole in your skull, and then we can stick a fiber optic cable in there. A true story. And we can shoot a laser, still a true story, through that fiber optic, and that light will spread just a little bit in your, not much, but just a little bit, just far enough to activate wherever we put these particular ion channels. And we can open and close ion channels which will either create or prevent action potentials in a select number of neurons in your brain. I think that's pretty exciting, right? Because then you could just like turn on and off parts of your brain with lasers. Yeah, it's pretty exciting, right? So that's actually, that technique's actually probably less than 10 years old. That's a pretty recent approach. The 2 guys who did that was this guy, I call him Big Carl because I think everybody named Carl, their nickname should be Big Carl. I don't know why, I just think that's, but this guy is pretty big. His name's Carl Dieseroth, and then Ed Boyden, who kind of came up with this idea a few years ago. It's pretty brilliant. They originally tried magnets. They thought they were going to pull ion channels open with little magnets, and that didn't work, Eric. So they actually thought, well, hey, what about light? It's actually based on this algae. They can get DNA from that. Anyway, kind of exciting. Don't worry about this neurochemical stuff. You can measure those chemicals. Again, that microdialysis or fast cyclic voltammetry, you can measure dopamine or any other neurotransmitter release. Not a big deal. Hey, who's got a twin? Really? Do they also go into the wrong classrooms? Probably. It's okay. So twin studies and adoption studies, these are pretty cool for genetic approaches. You guys know the difference between monozygotic and dizygotic twins? Yeah, 1 and 2, right? So monozygotic, you only had 1 zygote, and then you ended up being 2 people. So your DNA is identical, those are identical twins. Dizygotic, you had 2 zygotes, so you always have been 2 independent people. Those are going to be what we call fraternal twins, right? So they're not going to be identical. The way twin studies work is you convince a person to have twins. Takes a lot of work. And then you take those 2 twins and you send one to one family and one to another and see how they turn out. Yeah, man, that's how it works. That's not exactly how it works. How this typically works with twin studies is they will find, they can measure traits between monozygotic and dizygotic twins and see if there's 100% concordance rate or not. You can determine whether or not, or to what degree, their hereditary influences, right? They'll also do with adoption studies. They don't do these as much anymore. And the reason for that is most twins, if they are up for adoption, they'll be adopted together, right? Sort of older logic was it was better to split them, right? And send one twin to one adoptive family and another twin somewhere else. So like years later, they could have this major reunion on 60 Minutes. And right? Do you remember when that was like, you would see these things all the time? They don't do that much anymore, right? Because twins stay together. But what we lost from that, scientifically speaking, is the ability to compare identical twins that have been raised in completely different environments, right? And so if you have identical twins in different environments and they both develop the same disorders, they both have the same sort of neurological issues, then we know, or we can say with maybe some more confidence that that's a genetic issue and not a environmental influence, right? So we can kind of pull those things apart. So, I don't think anybody's going to try to bring back twin studies or adoption studies, right? Nobody's going to like, oh, I'm going to try to have twins and have somebody in another state adopt one of them and see if they turn out differently 40 years from now. Nobody's signing up for that. Not a big deal. What about genome? Not too much we need to think about there. Although there are going to be a number of studies that we talk about with mice that have been mutated, right? So we'll take their DNA. We sort of know a lot about the mouse genome. We'll be able to go in and adjust those genes. We can adjust their function up or down. We can delete a function or a deleted gene which will alter function. We can do all those things. And that way we can see, what effects those particular genes have on behavior or neurological disorders. We'll call those knockout mice. Anybody heard of a knockout mouse? You will at some point, Elizabeth. Don't worry about it. They are not into boxing. Like the MMA of mice. Nobody thought that was too funny. I did, but that's okay. I'm just thinking about these two mice like in an octagon, like fighting each other. Like, don't worry about this. Okay, so that's the end of methods. Anybody have any questions about research methods? Again, I'm not going to hit you too hard on that.